So good morning students. So in earlier classes of this protein and amino acid chemistry, so already we have discussed about the structure of amino acids and how many amino acids are there in nature, and what are the standard amino acids with the names and their structure. And we have already seen the how they have been classified, amino acids, how they have been classified based on the different ways. And we have already discussed about the properties of amino acids, few properties based on their amino group, few properties based on their side chain, and few properties are based on their carboxyl group. So already those we have already discussed. And few amino acids which not participate in the protein formation are called non-standard amino acid. So already we have seen the examples of non-standard amino acids and few amino acids. So already these are the list of the amino acids, means derivatives which are derived from amino acids. So like that, and coming to the next topic is, see here. So now I think you have got an idea about the structure of the amino acids, and what is there in the common structure of amino acids. There is alpha carbon is there. To the alpha carbon, one side there is amino group, and other side there is carboxyl group. Okay, see here, if you see the structure of all 20 amino acids, so what is the common feature, structural feature to be seen here? There is if you see the structure of all 20 standard amino acids, to the alpha carbon, one side there is amino group, and on the other side there is carboxyl group, and a third valency of the alpha carbon has been attached to hydrogen, and the difference of all these amino acids occur at their R represents the side chain. Now here these amino acids, when they are joined, that would result in the formation of peptides. So how they have been joined, amino acids, by a bond known as peptide bond. So two successive amino acids, they have been joined by peptide bond. How a peptide bond is formed already I have explained in the previous class. The alpha amino carbon of one the alpha amino group of one amino acid has been joined to alpha carboxyl group of another amino acid by losing the water molecule. The so formed bond, we can call it as a peptide bond. Okay, so two successive amino acids has been joined by a peptide bond. A peptide bond connects carboxyl group of alpha carboxyl group of one amino acid to the alpha amino group of the second amino acid by losing one water molecule. If two amino acids are joined, that would result in the formation of a dipeptide. Okay, if two amino acids are joined to form a dipeptide. So that means dipeptide is composed of how many amino acids? Two amino acids with one peptide bond. Like that, if three amino acids has been joined, that would result in the formation of a tripeptide. Tripeptide means there will be two peptide bonds are there, like that, so on, based on the number of amino acids. If four are there, tetrapeptide. If five are there, joined by peptide bond, result in the formation of pentapeptide. Like that, up to 50 these amino acids, if they are joined by peptide bonds, those you can call it as a peptide. Okay, up to 50. Okay, minimum 2 up to 50. If the amino acids, if the number of the amino acids are in the particular one, if the number of the amino acids are from 2 to 50, those you can call it as a peptides. Okay, 2 to 20 if they are joined by peptide bond, the so formed product you can call it as a peptide. So based on the number of amino acids you can divide like that. That is a tripeptide. If two are there, dipeptide. If three are there, tripeptide, tetrapeptide, pentapeptide. More than 10. Okay, if 10 are there, that is called. If 8 are there, that is called octapeptide. If 9 are there, nonapeptide. If 10 are there, decapeptide. More than 10 amino acids means 10 to 50. Okay, if the particular particular one has it 10 number of amino acids are from 10, more than 10. 10 means 11 to 50. Those you can call it as a polypeptide. Okay, polypeptide means the amino acids may be in between more than 11 to 50. Okay, so if the number of the amino acids are more than 50, those you can call it as a protein. Okay, so up to 50, based on the number of amino acids from, okay, up to 50, the so formed product you can call it as up to 50. If the amino acids joined by peptide bond, the so formed product you can call it as a peptide. If more than 50 are there, that those you can call it as protein. I think you understood that difference between a peptide and a protein. Both are made up of amino acids only. Then how do you differentiate whether it is a peptide or whether it is a protein? Means here based on the number of amino acids. Number of amino acids are less than 50. Those are called polypeptides, means peptides. More than 50 means those you can call it as proteins. Now even these peptides, less than number of amino acids less than 50 peptides has, these are biologically important. Means these polypeptides, means these different peptides exist in the nature, and these polypeptides are play an important role in the they have physiological role, and they play an important role in the biochemical reaction. Okay, now let us discuss about the biologically important peptides. This is an important short answer question for you. Biologically important peptides means the peptides, the biological importance, physiological role of the peptides. Okay, so first examples of the biological important peptides are carnosine and anserine. Okay, carnosine and anserine, these are the two dipeptides. Dipeptides means these two are made up of, these carnosine is a dipeptide and anserine is a dipeptide. These two are made up of two amino acids. That's what this, it's a dipeptide, and it is present in the skeletal muscle and also present in the myosin ATPase activator, and plays an important role in stabilizing the pH in the muscle cells. So these two are the dipeptides, carnosine and anserine. Where they are present in the skeletal muscle, mainly they activate the myosin ATPase. Okay, myosin ATPase, and then it helps in stabilizing the pH in the muscle cells. Then the next one is aspartame, biological important peptide. This is aspartame. It is also dipeptide made up of two, the name of the amino acids are aspartate and phenylalanine, and it is more sweeter than the sugar and honey, so it is used as artificial sweetener. Okay, so the second one is a aspartame. It's a dipeptide made up of aspartate and phenylalanine. It's an artificial sweetener used in the sweet industries. Okay, next one is the creatinine. Okay, next biological important peptide is creatine. Okay, it's not creatinine, please make a note, it is creatine. Creatine is a tripeptide, means how many amino acids are there in the tripeptide? There are 3 amino acids there in this creatine. And how many peptide bonds are there? There may be 2 peptide bonds with 3 amino acids. Name the amino acids which participate in the synthesis of creatine are simplest amino acid, that is glycine, basic amino acid arginine, and sulfur containing amino acid methionine. 3 amino acids are participating or involved in the formation of the creatine, that is a tripeptide. So here 2 major organs are involved in the formation of the creatine, that is liver and kidney. Okay, 2 reactions are there in the formation of creatine from these 3 amino acids. So first one reaction takes place in the 2 major organs are involved in the formation of the creatine, that is liver and the kidney. Now here once the creatine has been formed from the kidney, it is transported to the skeletal muscle. Okay, where in the skeletal muscle creatine is phosphorylated to creatine phosphate. Okay, please note down, creatine phosphate is a high energy phosphate compound. Okay, high energy phosphate compound means creatine phosphate, which is the reserve storage of energy in the muscle. Means breakdown of the creatine phosphate on hydrolysis yields more than 7 calories of energy. Okay, what do you mean by high energy compounds? High energy compound means ATP is one of the example of high energy compound. Meaning of energy compound is if that compound on hydrolysis means on breakdown, if it yields, if it generates more than 7 calories of energy, that compound is said to be high energy compound. One of the example of energy compound is creatine phosphate. What is this creatine phosphate? First, creatine is formed from 3 amino acids, glycine, arginine and methionine. The so formed creatine in the skeletal muscle, it is phosphorylated to creatine phosphate. Creatine phosphate is the reserve energy for muscle contraction. So whenever muscle needs energy during the contraction, then it is provided by hydrolysis of this breakdown of this creatine phosphate. So it provides energy during muscle contraction. That is the importance of this creatine. That is it. One of the biological important peptide. It's a tripeptide. Next, coming to other important peptides, that is TRH. It's a hormone. Okay, which hormone? TRH stands for thyrotropin releasing hormone. Okay, thyrotropin releasing hormone. It's a tripeptide. Okay, and this thyrotropin releasing hormone, it stimulates. Okay, what are the 3 amino acids which are present in this thyrotropin releasing hormone? Means it is also known as prolyl, glutamyl, histidine, proline. Okay, so the 3 amino acids are proline is there, glutamate is there, histidine is there in the structure. Okay, so TRH is a tripeptide. The 3 amino acids which participate here in the formation of the TRH is proline, histidine and glutamate. These 3 amino acids joined to form a hormone known as thyrotropin releasing hormone. And what is the function of this thyrotropin releasing hormone? It stimulates the anterior pituitary to release the thyroid stimulating hormone. Okay, thyrotropin releasing hormone stimulates the anterior pituitary to release TSH, that is thyroid stimulating hormone. Now, thyroid stimulating hormone in turn stimulates the thyroid gland to release thyroid hormones T3 and T4. T3 and T4, that is triiodotyrosine and thyroxine. These two are thyroid hormones, and you know we all know that thyroid hormones has function in each and every cell of the body, and they mainly regulates the body temperature. So this thyrotropin releasing hormone is also a tripeptide. Now next one is oxytocin. Oxytocin is a nanopeptide. Nanopeptide means how many amino acids are there? 9 amino acids are there, and this oxytocin is released. Okay, it is secreted from the posterior pituitary gland, and we know that this oxytocin, it stimulates the contraction of the uterus. Okay, during the delivery labor time, this oxytocin is induced to induce the contraction of the uterus. Okay, function of the oxytocin is it stimulates the contraction of the uterus, so hence it is used to induce the labor. So now oxytocin is nothing but it's a peptide. How many amino acids are there? 9 amino acids are there. Then coming to the other important biological important peptide is vasopressin. Vasopressin is also known as ADH. ADH means antidiuretic hormone, and it is also a nanopeptide made up of 9 amino acids, and this vasopressin is secreted from posterior pituitary gland, and the function of the antidiuretic hormone is, we know that it helps in the reabsorption of the water from the renal tubules. Okay, see here, antidiuretic hormone, it helps in the reabsorption of water from the renal tubules. So hence it helps in the maintenance of the water and electrolyte balance. That is the function of the vasopressin. Antidiuretic hormone, if there is deficiency of the ADH is there, there is polyuria. Polyuria means increased urinary output is there because lacking deficiency or decrease of the reabsorption of the water from the renal tubules. Now, this vasopressin, which plays an important role in water and maintenance of the water and electrolyte balance, it is also a nanopeptide, biological important peptide. And the next one is angiotensin 1 and angiotensin 2. These two are also a peptides. Angiotensin 1 is a decapeptide. It has 10 amino acids are there, and angiotensin 2 is a octapeptide and it has 8 amino acids are there. And both angiotensin 1 one and angiotensin, renin-angiotensin mechanism, which maintains the blood pressure. Okay, so this angiotensin 1 and angiotensin 2 participate in the renin-angiotensin mechanism and plays an important role in the regulation of the blood pressure. And this angiotensin, both 1 and 2, are also nothing but peptides. Now, the next biological important peptides are few antibiotics. Few antibiotics are also peptides. Like examples are gramicidin, actinomycin, and polymyxin B. So these are also these are antibiotics widely used in like antibacterial drugs, mainly so used to treat the bacterial infections. Examples are gramicidin, actinomycin, polymyxin B. All these are also naturally occurring peptide antibiotics. And next one, gastrointestinal hormones, namely gut hormones. So GIT hormones, namely gastrin, cholecystokinin, and secretin. All these are peptide hormones. Okay, so these hormones plays an important role in digestion and absorption of the nutrients. Okay, gastrointestinal hormones like gastrin, cholecystokinin, and secretin, all these are nothing but a peptide hormones, and we know that plays an important role in digestion as well as absorption of the nutrients like carbohydrates, proteins, fatty acids, fats, etc. The next one is other biological important peptide is bradykinin and calidin. Okay, please note down, this is the important question. So what we are discussing is biological important peptides, okay, with their importance, with their biological function. And the next one is bradykinin and calidin. Okay, bradykinin is a nonapeptide made up of 9 amino acids, and calidin, okay, it's a decapeptide. So the main function of this bradykinin and calidin is they are powerful vasodilators. So vasodilators means they cause the muscle relaxation. So because of that, they may dilate the blood vessels and helps in maintaining the blood pressure. Okay, both these are the bradykinin and the calidin or the peptides. They are the vasodilators, dilate the blood vessels and regulate the blood pressure since they are muscle relaxants. And the next one is the other important biological peptides are enkephalins. Enkephalins are pentapeptides. So penta means made up of 5 amino acids. So these are naturally occurring painkillers in our body. Okay, enkephalins are the peptides. So how many? Made up of 5 amino acids. So these are the painkillers. So our body has its own mechanism for control of the pain by secreting this enkephalins. These are nothing but peptides. Okay, and the next one, other examples of the biological important peptides are the hormones. Glucagon. Glucagon is a hormone. How many amino acids are there? 29 amino acids are there, and glucagon is secreted from beta cells of islets of longer hands of pancreas. Okay, so glucagon is secreted, produced by the pancreas, and the glucagon is a hyperglycemic hormone. Okay, hyperglycemic means glucagon elevate the blood glucose levels, increases the blood glucose levels. So when there is a fall in the blood glucose level less than the normal, hypoglycemic condition. Hypoglycemia means when your blood sugar level is decreased, falls less than the normal level. So that hypoglycemia, because of the hypoglycemia, brain will be affected because brain needs the continuous production of the continuous energy, but brain mainly depends on glucose for the energy. Okay, the food for brain is only glucose. So if there is fall in the blood glucose levels is there, the first immediately brain will be affected. So immediately that decreased glucose levels will be sensed by the glucose, and it increases the blood glucose levels and bring the glucose levels to the normal levels. Why? Because glucagon, what is the action of the glucagon? It is a hyperglycemic hormone. And even there is another hormone is there, insulin. Insulin has 51 amino acids. Okay, in the glucagon how many amino acids are there? 29. Even from the pancreas another hormone is secreted, that is insulin. Insulin is made up of 51 amino acids. And what is the function of insulin? Insulin is a hypoglycemic hormone. Hypoglycemic hormone means it decreases the blood glucose levels. What about the glucagon? Glucagon elevates, increases the blood glucose level. So here the homeostasis of the blood glucose level is constantly maintained in the blood by balancing between the secretion and production of the insulin and glucagon. Please note down both insulin and glucagon are the peptide hormones. Insulin, okay, when there is hyperglycemia, blood glucose level is more than normal, which will be released into the circulation, insulin. When there is fall in the blood glucose level, that is hypoglycemia, in response to that, when hypoglycemic condition stimulates the release of the glucagon. Okay, so these two are the peptide hormones. Now here the next one is ACTH, that is adenocorticotropic hormone. It is made up of 39 amino acids are there. It is mainly produced and secreted by anterior pituitary. This ACTH, adenocorticotropic hormone, produced and secreted by anterior pituitary, and mainly it increases the production and release of the corticosteroid hormones by the cortex of the adrenal gland. The function of the ACTH is it stimulates the production of cortisol from the cortex of the adrenal gland. And the next one is here glutathione. Next important biological important peptide is glutathione. Glutathione is a tripeptide. How many amino acids are there? Tripeptide means we know it is made up of 3 amino acids. Name the amino acids which participate in the glutathione is called glutamate, cysteine and glycine. Hence it is known as glutathione is also known as gamma glutamyl cystinyl glycine. Okay, glutathione, as it is a tripeptide, 3 amino acids participate in the formation of the glutathione. The name of the amino acids are glutamate, cysteine and glycine. So glutathione is also known as gamma glutamyl cystinyl glycine. And this glutathione is found in almost, sorry here it's a spelling, it's a typo error. It is found in almost all the cells except the neurons. Okay, see here this glutathione is a tripeptide present in almost all the cells except the neurons. The main function of the glutathione is it acts as an antioxidant. Okay, glutathione acts as an antioxidant and it scavenges the free radicals from the body. It removes the free radicals. If free radicals are there, they may damage the cell membrane. So due to the biochemical reactions in the body, there is release of the free radicals are there, and these free radicals are highly reactive. They may damage the cell membrane. So time to time these free radicals has been scavenged, removed from the body by this glutathione. So it protects the cell membrane. So it acts as an antioxidant. So mainly here this H2O2 is hydrogen peroxide. Hydrogen peroxide is produced in the body due to various metabolic reactions in the body, but hydrogen peroxide is toxic to the cell membrane and toxic to the RBC membrane. Okay, that means if H2O2 is remains like that in the blood, it may rupture, it may damage the cell membrane and RBC membrane. So this the body H2O2 production cannot be stopped because it is produced due to various metabolic reactions, but H2O2 which is produced in the body has been removed from the body by this glutathione. Okay, so glutathione plays an important role in the scavenging the free radicals or in the detoxification of hydrogen peroxide, thereby protecting the cell membrane, or it prevents the cellular damage. And it also maintains, as H2O2 is detoxified by using the glutathione, it maintains the RBC membrane integrity and it also protects the RBC membrane. Okay, see here how this glutathione prevents this H2O2 means usually here this glutathione in the blood, it exists in two forms, that is oxidized form and reduced form. Listen carefully. Glutathione in the blood, it exists in two forms. Reduced form is GSH and oxidized form is GSSG. Okay, in the two forms it exists. The reduced form of glutathione, that is GSH, plays a major now here that help in the that keeps the iron in the Fe+2 state. Okay, glutathione reduced form, that is GSH form, which keeps the iron of the hemoglobin in Fe+2 state, thereby maintains the RBC membrane integrity. If you remember, in the hemoglobin, iron is there. Iron is always present in Fe+2 state. So that Fe+2 state means ferrous state. It is constantly maintained by this glutathione. So thereby it maintains the RBC membrane integrity. Now, the oxidized glutathione is there, that is GSSG. It detoxifies H2O2, hydrogen peroxide, by converting into water. Okay, toxic hydrogen peroxide is converted into water. In that conversion, oxidized glutathione, that is GSSG, is converted to reduced glutathione. Okay, like that, glutathione plays a major role in the detoxification of H2O2 and thereby protecting the RBC membrane and cell membrane, and it protects the cell from the damage. Hence, it acts as an antioxidant. Okay. Now here, even this glutathione, it serves as coenzyme for prostaglandins, and these are the growth factors, and even this gamma glutamal cystinyl glycine, that is glutathione. Okay, that is Mr. cycle, which help in the absorption and transport of amino acids from the gut. That means glutathione not only is scavenging the free radicals, antioxidant, which help in the absorption and transportation of amino acids from the GIT, and it has a role in the detoxification, and it helps in stabilizing the disulfide linkages in the protein structure. Okay, disulfide linkage in the protein structure has been stabilized by this glutathione, and it also keeps the iron of the hemoglobin in ferrous state. Okay, so these are the main functions of the glutathione, one of the biologically important peptide, and it's a tripeptide. Name the amino acids which participate in the synthesis of the glutathione, that is glutamate, cysteine and glycine, and it protects the RBC membrane integrity, cell membrane integrity, prevents the cellular damage, acts as an antioxidant, provides, and it helps in absorption of the and transportation of amino acids, has a role in the detoxification. Okay, that's it about the biologically important peptides. Okay, biologically important peptides. Name the biologically important peptides: carnosine, anserine, aspartame, creatine, TRH, that is thyrotropin releasing hormone, oxytocin, ADH, that is vasopressin, angiotensin 1 and 2, peptide antibiotics, GIT hormones, that is gastrin, cholecystokinin, secretin, etc., bradycardine and calidin, enkephalins, glucagon, insulin, ACTH, glutathione. All these are biologically important peptides. Now here, if more than 50 amino acids has been joined by a peptide bond, that would result in the formation of a protein. Now let us discuss about the there are different 100 different proteins are there in the nature. So those proteins, okay, different proteins has been classified into various types. Okay, now let us discuss about the protein classification. Proteins means made up proteins are the large molecules with a definite size and shape, and proteins are the charged molecules. Okay, proteins carries either positive charge or negative charge, and the elements which participate in the structure of the protein are carbon, hydrogen, oxygen and nitrogen are the major. Along with that, there may be sulfur and phosphorus, etc. are there. Okay, now these proteins which are which exist in the nature has been classified into various ways. So there are 4 different classifications of the proteins are there. So first classification of the protein is based on its function. Second one is based on its solubility. Based on the solubility and the chemical nature, classification is made, and other type of classification is based on the shape, and proteins are classified based on the nutrition. Okay, there are 4 types of classifications are there: based on the function it performs, based on the structure, based on its shape, and based on the nutrition. Now coming to the classification of the proteins based on the function of the protein. So few proteins which has catalytic function, Okay, catalytic function means which catalyzes the biochemical reactions. Those proteins you can call it as enzymes. Okay, proteins which has catalytic function, examples are enzymes. And structural proteins, few proteins are the components of the structure, that is collagen, elastin and keratin. These are the structural proteins. And few proteins has a role in the contraction muscle. Okay, that is myosin, actin, flagellar proteins. These are the contractile proteins. Few proteins help in the transportation. Those are called transport proteins. Example is hemoglobin, myoglobin, albumin, transferrin, etc. Hemoglobin help in the transport of gases, oxygen and carbon dioxide, and albumin help in the transport of the some compounds, especially insoluble in blood, has been transported in the blood along with the albumin. Transferrin is a transport protein, help in the transfer of iron. Next one is regulatory proteins or hormones like ACTH, insulin, growth hormone. All these are the hormones which has a role on the metabolic pathways, which regulates the metabolic pathways. And few proteins are there, genetic proteins. Example is histones. Histones are mainly found in association with the nucleic acids. Now here protective proteins, like a few proteins protects the body from the infections. Examples is immunoglobulins, interferons, and few proteins has a role in the clotting of the blood. Okay, protect the that is almost all blood clotting factors are also nothing but proteins. So like that, proteins are classified based on the function, like catalytic proteins, structural proteins, contractile proteins, transport proteins, regulatory proteins, genetic proteins and protective proteins. Then coming to the next one, second one, classification based on the composition and solubility. Okay, second classification is based on the composition of the protein and its solubility. Proteins are broadly classified into three based on its composition and solubility. What are the three subclasses? Three classes based on the composition and solubility is simple proteins, conjugated proteins and derived proteins. Okay, based on the composition and its solubility, proteins are classified into three major classes, that is simple proteins, conjugated proteins and derived proteins. Let us discuss about each one. First, simple proteins. Why they are called simple proteins? Because as per the definition, they contain only amino acids. No other additional groups are there. Only amino acids are there. Okay, if simple proteins are composed of only amino acids. Examples of the simple protein is albumin. Albumin is a simple protein. Means if you see the structure of the albumin, it is composed of only amino acids. And these albumins are soluble in water. Okay, and these are heat coagulable protein. Okay, when exposed to heat, albumin is coagulated. So albumin is soluble in water and it's a heat coagulable protein. Albumin is mainly present in the serum. Okay, serum, important plasma protein is albumin. Important plasma protein is albumin because more than 50% of the plasma protein is constituted by albumin. Albumin contributes more than 50% of your protein, your plasma protein. And albumin, as it constitutes more than 50% of the total plasma proteins, albumin mainly helps in maintaining the colloidal osmotic pressure. The main important function of the albumin is it maintains the colloidal osmotic pressure. Maintenance of the colloidal osmotic pressure means this albumin, it helps in exchange of the fluids between intracellular and extracellular fluids. Okay, the fluids, how they'll be exchanged between intracellular and extracellular means with the help of albumin. Thereby it exerts the osmotic pressure. Next, albumin, it acts as a buffer, helps in maintaining the blood pH. And albumin, it helps in the transport of the substances, especially which are insoluble in blood. Example is unconjugated bilirubin, free fatty acids. So they will be transported in the few minerals like copper. How they'll be transported in the blood means along with the albumin. Albumin helps in the transport of the compounds, especially which are those insoluble in water. Example, unconjugated bilirubin, free fatty acids, etc. And even albumin help in the transport of the drugs to their target organs. So these are the main functions of the albumin. So that albumin is a simple protein. It's a heat coagulable protein, and it's a low molecular weight protein. Its molecular weight is only 69,000. It's a low molecular weight protein, but here it's the important plasma protein. Okay, and you know the functions of the albumin. It is made up of only amino acids, and not only in the serum, even albumin is present in the egg. Okay, that is ovalbumin, egg. Okay, egg albumin it is also present in the egg, that is egg albumin, and even it is also present in the milk, that is lactalbumin. Okay, serum albumin in human blood and egg albumin in the egg, lactalbumin in the milk. These are the various albumins. Then the next one, simple protein, is globulins. So globulins are insoluble in pure water, but they are soluble in dilute salt solution. What about the albumins are soluble in water, whereas globulins are insoluble in pure water, but where they will be soluble in dilute salt solution. And even a globulin is also a heat coagulable protein. So heat coagulable proteins are albumin and globulin, but albumins are soluble in water, globulins are insoluble in water. And even the globulins are also present in the blood. Okay, albumins and globulins are important plasma proteins. Albumin and globulins are important plasma proteins, together form the total protein. Albumin and globulin both are simple proteins, important plasma proteins. Okay, now, and both are heat coagulable proteins. And even globulin is also present in the legumin of peas, and it was also present in the egg. And the next one is other simple protein is protamines. So protamines are soluble in water, and these are even soluble in the dilute acids and alkalis. Next simple protein is a protamines. Protamines are soluble in dilute acids, alkalis and water, and these are not heat coagulable protein. Albumin and globulin are heat coagulable protein, but protamines are not heat, where they are not coagulated by heat. And the protamines are strongly basic in nature because they are rich in basic amino acids, arginine and lysine. Protamines are strongly basic because they are rich in basic amino acids, arginine and lysine. As they are strongly basic, they are readily combined with the acidic proteins. Okay, these protamines, as they are the strongly basic in nature, they are readily associated with the acidic proteins. And even as they are the negatively charged, they are readily associated with the nucleic acids. Now here the proteins are strongly basic. They are readily associated with the negatively charged nucleic acids. Now these protamine zinc insulinate. Okay, protamine zinc insulinate is the common commercial preparation of the insulin. Okay, that's it about the protamines. And the next one is prolamines. Prolamines are one of the simple protein. They are soluble in 70 to 80% of the alcohol. Okay, they are soluble in organic solvents, like 70 to 80% of the alcohol, insoluble in pure water. And prolamines are rich in proline, and but they are lacking the lysine. Prolamines are rich in proline, deficient in the lysine. Examples of the prolamines are zein of corn. Okay, zein, that is a protein which is present in the corn, that is deficient in lysine, rich in proline. And even the gliadin of the wheat. Okay, gliadin of the wheat is also rich in proline, deficiency of the lysine, and hordein of the barley. And these are all the prolamines. So prolamines means, you just remember, these are the ones which are soluble in organic solvents, insoluble in water, rich in proline, and deficient in lysine. Example is zein of corn and gliadin of the wheat. And prolamines is a simple protein. And the next simple protein is lectins. Lectins means these are the proteins having high affinity to the sugar groups. Okay, they have high affinity towards the carbohydrates, and these are widely distributed in nature, but mostly they are derived from the plant sources, but also they may be present in the animals. Okay, but mostly they are derived from plant sources, and the main property of the lectin is they have high affinity towards the carbohydrates, towards the sugar, and these lectin from the dolichos biflorus that will agglutinate the human blood group A1 RBCs. Please note down lectins from this dolichos biflorus which agglutinate the precipitate the human blood group A1 RBCs, and the dolichos lectin will bind specifically with the alpha D N-acetyl galactosamine. So where it will bind means especially it binds to the alpha D N-acetyl galactosamine. It's a carbohydrate mucopolysaccharide. Now here there is a PHA. PHA is phytohemagglutinin. Okay, phytohemagglutinin. It is the lectin which is derived from the red kidney bean that will agglutinate all RBCs and WBC and produce mitogenesis of lymphocytes. Okay, there is a lectin. One of the lectin is PHA, that is phytohemagglutinin, that is derived from this red kidney bean that can agglutinate, precipitate the RBCs and WBC and produces mitogenesis of the lymphocytes. So these lectins are also simple proteins, and even lectins, they act as the receptors on the cell for recognition of galactose residues of glycoproteins. As I said, they have the high affinity towards the carbohydrate, so they act as the receptors on the surface of the cell for recognition of the galactose residues of the glycoprotein, and for hydrolytic, and even it acts as the receptors for hydrolytic enzymes containing mannose 6-phosphate, and it acts as and target these enzymes to the lysosomes. So that means lectins which are present on the surface of the cell help in the recognition of the galactose, and it also help in the recognition of the mannose 6-phosphate by the hydrolytic enzymes. So that means it makes the enzyme function easier. Okay, that is the functions of the lectins. And another simple protein is scleroproteins. Scleroproteins are insoluble in water, salt solutions and organic solvents, and soluble in hot strong acids. Okay, and the scleroproteins mainly they form the they help in the alignment and supporting the tissues. Examples of the scleroproteins, mainly these are present in the connective tissue, like their collagen of the bone, cartilage and tendon, keratin of the hair, keratin which is mainly present in the hair, horn, nails and hooves. Okay, so scleroproteins examples are collagen and keratin, connective tissue proteins, or scleroproteins. Scleroproteins are also comes under simple proteins. Okay, now here that's it about the simple proteins. In the simple proteins also these albumin, albumins, globulins, protamines, prolamines, lectins are globular proteins. Okay, the first 5 are globular proteins. They are either spherical or oval in shape, and one of the simple protein, that is here it is scleroprotein. These are fiber like structure, rod like structure, so it is a fibrous protein. So these 6 are the examples of the simple proteins. What do you mean by simple proteins? These are made up of only amino acids, but examples are albumin, globulins, protamines, prolamines, lectins, scleroproteins, etc. Then coming to the next one is conjugated proteins. So conjugated proteins means these are the proteins in combination, okay, with the non-protein part. Protein part is there, along with the protein, non-protein part is there. The non-protein part of a protein is called prosthetic group. The non-protein part of a protein is called prosthetic group, and if the protein plus non-protein part together are present in a protein, that is said to be a conjugated protein. Remember, the non-protein part is called prosthetic group. Examples of conjugated proteins are glycoproteins. Glycoproteins means carbohydrates in combination with the protein. Means proteins in combination with the carbohydrate. So where these carbohydrates has been attached to the protein means especially serine and threonine. Which type of amino acid? These two hydroxyl amino acids. So hydroxyl groups of serine and threonine And amide groups of asparagine and glutamine, they form linkage with the carbohydrate. Okay, glycoprotein is a conjugated protein. What do you mean by conjugated protein? Protein part along with the non-protein. Okay, and in the glycoprotein is a combination of protein with the carbohydrate. Now name the prosthetic group which is present in the glycoprotein. Prosthetic group in the glycoprotein is a carbohydrate, right? So carbohydrate is a non-protein part. So where this carbohydrate has been attached to protein means especially the hydroxyl groups of serine and threonine or amide groups of asparagine and glutamate, they may join with of these proteins, amino acids join with the carbohydrates to form a glycoprotein. Examples of most of the blood proteins, serum proteins are glycoproteins, and blood group antigens are also glycoproteins. Okay, blood group antigens and many serum proteins are the examples of glycoprotein. Means the proteins which are present in the serum are most of them are present in combination with the carbohydrate as glycoprotein. Okay, right? And here if the carbohydrate content is more, okay, sometimes glycoproteins, if the carbohydrate content is more, so those result in the formation of mucoprotein, okay, or proteoglycans. Try to differentiate glycoprotein and proteoglycans. Glycoprotein means protein in combination with the carbohydrate. Proteoglycans means proteins in combination with the carbohydrate only. Then how do you differentiate glycoprotein and proteoglycan? Means based on the carbohydrate content. If the carbohydrate content is less than 10%, 4%, that is said to be glycoprotein. Okay, carbohydrate content is less than 4%. The so formed conjugated protein, you can call it as glycoprotein. If the carbohydrate content is more than 10%, the so formed conjugated protein, you can call it as proteoglycans, or else you can call it as mucoprotein. Okay, I think you understood here the difference between the glycoprotein and proteoglycans. Both are conjugated proteins only. But how glycoprotein or proteoglycans, both are made up of carbohydrate in combination with the protein. But how do you differentiate glycoprotein and proteoglycan? Based on the carbohydrate content. If less carbohydrate, less than 4% of the carbohydrate is there, the so formed conjugated protein is called glycoprotein. Most of the serum proteins, blood group antigens are glycoproteins, right? If the carbohydrate content is more than 10%, if more carbohydrate content is there in combination with the protein, the so formed conjugated protein is called proteoglycan or mucoprotein. As the carbohydrate content is more, proteoglycans are viscous in nature. Okay, as the carbohydrate content is more, they are viscous in nature. The viscosity helps in it, as they are mainly proteoglycans are present in the a fluid, synovial fluid around the joints. So there it acts as shock absorbent and lubricant. Okay, I think you are following. Glycoproteins are serum proteins. Most of the serum proteins are glycoproteins. Here the carbohydrate content is less. If carbohydrate content is more, that would result in the formation of mucoproteins or proteoglycans. As the carbohydrate content is more, they are viscous in nature. Because of the viscosity, they are mainly present in synovial fluid of the joints and acts as shock absorbent and lubricant. So this is one of the conjugated protein, and rest of the conjugated proteins classification will continue in the next class. Thank you.